Hi, my name is Shreya Krishnan, and in this video, I will be talking about shape analysis of sickle cells via the LCPC transform and how it reveals distinct shape states invisible to area. The LCPC transform measures the shape of objects. In biology, the structure of an object determines its function. Therefore, measuring the structure of an object gives us information about its function. In the case of sickle cells, the LCPC transform measures precise variations of sickle cell shapes in a way that provides insights into response versus non-response to drugs and into the effects of gene editing. To perform the LCPC transform, first, a mask of the outline of the object of interest is created, which, in this project, is a sickle cell. In step 2, a parallel grid is overlaid onto the outline. The third step is to calculate the distance from the baseline of the parallel grid, shown as a vertical dashed line, to the intersections between the parallel grid and the outline of the object. The fourth step is to linearize the parallel grid system. If one line intersects the outline of the object multiple times, the distances from the baseline of the grid to each intersection along one line of the parallel grid are summed. A graph of the distances is made. Each line on the parallel grid is shown on the x-axis in the step 4 graph, and the distance is shown on the y-axis. The fifth step is to do a fast Fourier transform on this data to create a frequency plot that is a multi-dimensional representation of the sickle cell shape. In this presentation, the fast Fourier transform is abbreviated to FFT. On each graph, the x-axis represents the order of frequency bins. Each bar represents the value of the magnitude for the corresponding frequency bin. In this presentation, the bin number is the same as the bar number. In the sixth step, a frequency plot can be converted to a scalar value that represents the shape. For example, all bins, or bars, can be summed. First, the LCPC transform was applied to phantom sickle cell shapes. Applying the LCPC transform to sickle cell shapes reflects the shape itself. The cell image on the left, the holly leaf variant, has multiple concavities. The FFT plot for this cell forms a bell curve in the middle bins, as shown by the red line on the graph. In contrast, the normal cell is round and convex. Its shape can be considered the inverse of that of the holly leaf variant. The FFT plot for the normal cell reflects this relationship, as there is a trough in the middle bins for the normal cell, which is outlined in red on the graph. The shape formed by the first 3 bins, which are boxed in black, can indicate the curvature of sickle cells. In sickle cells 1 and 2, shown on the left, the first bin is high, the second bin is low, and the third bin is in between the heights of the first and second bins. This pattern, outlined in red, forms a shape similar to a ladle. In sickle cells 3 and 4 on the right, this ladle pattern disappears, and the first 3 bins form a hockey stick shape, outlined in green. Visually, the curvature of sickle cells 3 and 4 is less. So, the pattern formed by the first 3 bins is correlated to the sickle cell curvature. Another important trend found in this set of sickle cell drawings is the changes in FFT plots across different types of sickle cell shapes, particularly in the middle bars, which are boxed in red. In the S shape, the middle bars curve slightly instead of remaining flat as they did in the crescent. In the diamond shape, these middle bars form a clear upward curve. The differences in these highlighted bars are good candidates to create an index to classify these shapes, and creating gradient themes containing phantom images where only one condition is varied gives further insight into the FFT plots of these sickle cell shapes. In the first gradient theme, the right side curvature of the crescent shape, which is outlined in red, was changed. In this gradient, the ladle pattern in bins 1 to 3, which are boxed in red, changed as the cells became thinner, or as the right side became more concave. The LCPC transform revealed something not visible by direct observation, which was an inflection point in shape 4. At this point, bins 2 and 3 become almost equal in height. After shape 4, the ladle pattern disappears and is replaced by the hockey stick pattern. This inflection point at shape 4 indicates that a new peak forms between shapes 3 and 4 that disrupts the smooth increase in magnitude between bins 2 and 5 that was seen in shapes 1 to 3. In the plot of the ratio of bin 2 over bin 3, shown on the left, the ratio increases across the gradient and is very close to 1 for shape 4. There may be some perfect sickle cell shape between shapes 4 and 5 where the ratio of bin 2 to bin 3 is 1. In the index on the right, the height of bin 1 decreases as the thickness of the shape decreases. Importantly, there is a plateau in values in both indices for shapes 5 and 6. This demonstrates how multiple scalar indices can point out similar patterns across shapes. In the second gradient theme, the right and left side curvatures of the crescent shapes were changed. All graphs for this gradient theme are shown here. In this theme, the bump in the middle bins, which is boxed in red, becomes less prominent as the curvature of the sickle cell shape decreases. Since this trend was not present in theme 1, where the right side shape was altered, this indicates that the left side shape affects the curvature of the middle bins. Also, in these shapes, the height of bin 1 decreases at a different rate than the height of bin 14, or the middle bin. An index can be created based on this observation. For all shapes except shape 2, the ratio of bin 1 to bin 14 increases as the thickness decreases, or the height of bin 14 is decreasing faster than the height of bin 1. This ratio points out that shape 2 has an equilibrium point, which causes it to stand out. This gradient also has an index that can be used for situations requiring a linear relationship. All shapes in this gradient theme exhibit the ladle pattern in the first 3 bins. But in this gradient, the height of bin 2 decreases at a faster rate than bin 3. This causes the ratio of the height of bin 2 over bin 3 to decrease as the curvature decreases, which is shown in this graph. In the third gradient theme, the thickness of diamond shapes was varied. All graphs for this gradient theme are shown here. In this gradient, the thin shapes, shapes 1 to 3, have high-frequency sinusoidal FFT plots. By contrast, the thick shapes, shapes 5 to 7, have smoother FFT plots. Within this gradient, there are 4 distinct states, and the FFT plot shape for each state is outlined in a different color. Shape 1 has a high-frequency open book pattern with 2 peaks, which is outlined in pink. Shapes 2 and 3 have a high-frequency pattern with multiple troughs that is outlined in red. Shape 4 has a smooth open book FFT plot outlined in orange with a very low value for bar 14. Shapes 5, 6, and 7 all have an FFT plot shape that resembles a bird in flight, outlined in green. So, in this gradient, there is a threshold between shapes 3 and 4 and a threshold between shapes 4 and 5 where the state changes. Although all the shapes appear visually similar, the LCPC transform reveals patterns that offer more detailed insight into each shape. This is an example of what area simply cannot assess because shape is related to, but distinct from, area. In this gradient theme, scalar indices demonstrate the differences between states. Both the sum of bins 11, 14, and 17, shown on the left, and the ratio of bin 14 to the sum of bins 11 and 17, shown on the right, quantify the differences between states. In both indices, shape 1 stands apart from shapes 2 and 3. Shapes 2 and 3 have close values. Then, shape 4 exhibits a sharp change in height. In the left index, the height increases, and in the right index, the height decreases for shape 4. Shapes 5, 6, and 7 then form a linear trend on the left index and remain close in value in the right one. Also, in the index on the right, Each state has a distinct value range. In the fourth gradient theme, the thickness of S-shapes was varied using an S-shaped sickle cell drawing. All graphs for this gradient theme are shown here. There are 3 distinct states in this gradient that cannot be visibly observed, but are revealed by the LCPC transform. The FFT plots of shapes 1 to 3 all have an open book shape, outlined in red. The FFT plots of shapes 4 and 5 have a bowl shape, outlined in pink. The FFT plot of shape 6 has a mountain range shape, outlined in orange. There is a threshold between shapes 3 and 4 and between shapes 5 and 6 where the state changes. More refined shapes can reveal these thresholds. The LCPC transform measures precise variations of sickle cell shapes in a way that provides insights into response versus non-response to drugs and into the effects of gene editing. For consulting services and licensing, please reach out to BrainScanology at info@brainscanology.com. Thank you for watching this presentation.